In this section, we're going to go through some of the models having to do with the reproductive system as well as some of the reproductive histology. Starting with some of the activity plaques, we can see that as commonly with these, we've got both a macroscopic section and a microscopic section. So as you're going through and doing the reproductive anatomy, make sure that you're paying attention to the microscopic anatomy. In this case, we have the oocyte with the different layers around it. We've got the cumulosophorus way out here, this cloud of cells. We have just this first layer of cells here, which is the corona radiata, and then the pellucidum right here, which in this model is actually represented in green, but when you see it in the actual slide, it will be pink. Same with the male reproductive plaque, noticing the different parts of the sperm cell, although those are pretty simple. This ovary model is one that we don't have listed in the lab manual, but it is on the list of things to know for Lab Practical 3. We've got a couple different regions to it. We've got the cortex out here where we see all the follicles, and the medulla out here where we see the blood vessels. So the follicles are going to be found out in the cortex. And with this model, what they try to show you is a progression, a developmental progression of the follicles. Now remember that the follicles are going to be the oocyte that we see like here in brown, in this case like with a primary follicle. So it includes the oocyte in the center as well as the follicular cells around the periphery. So we've got different stages. We've got the primordial follicles here. These are the ones that are basically in stasis. We've got primary follicles basically here and here, where we see the oocyte is much bigger, and now the surrounding follicular cells are also much larger. And then we have a secondary follicle here. Now this secondary follicle, it's the best example of it in this model, but it also serves as what we call the mature follicle. We can see the oocyte is being released from the surface of the ovary, so this is actually ovulation occurring as well. This big hole, if you will, here is called the antrum, the fluid-filled antrum. And so really, we tend to use the presence of the fluid-filled antrum as our characteristic that we are looking at a secondary follicle, one that has developed from a primary follicle. It's kind of debatable as to when you get a secondary follicle, if you have multiple layers of follicular cells and then just the beginning of an antrum, then they graduate and say that that is a secondary follicle. But we would basically in the slides really focus on the presence of the antrum to identify a secondary follicle. Now, when you have multiple secondary follicles, as you commonly do, it's the largest secondary follicle that also then gets the name of mature follicle, also known as Graafian follicle. So after the egg is released, these surrounding follicular cells actually turn into this structure over here, which is called the corpus hemorrhagicum, because it has some hemorrhaging in the inside. We see some blood commonly on the inside. This corpus hemorrhagicum turns into a corpus luteum, or yellow body like you see here. So these again are the follicular cells that have now turned into a solid ball of cells that actually have its own function in terms of maintaining progesterone levels and maintaining the uterine lining. So this is the corpus luteum, and ultimately this will actually turn into a white body, which is also called the corpus albicans. We'll get into other developmental models a little bit later on, so we'll kind of skip forward past those. This is the menstruation model. I would say with this one, really, if you could identify the major components of the internal female reproductive tract, like we see here, or in any of these areas, like the fimbriae, the infundibulum, the fallopian tube or the uterine tube, the different parts of the uterus, different layers of the uterus, that's fine. I mean, this is supposed to represent the uterus going through different parts of the menstrual cycle, but they're really not that different. So identifying the parts of the different internal female reproductive anatomy, and then also this middle section here. So these two on the sides represent, much like our ovarian model that we just went through, different developmental stages for the follicle. So again, primordial and primary, secondary ones here, ovulation occurring right here, formation of the yellow body, or the corpus luteum, and then the corpus albicans. Same thing we're seeing right here as well. So I would say know the middle part of this model, and then at least one of these representations of the internal female anatomy. Again, a developmental one that we'll hit later on. We have quite a few in this last section. And then the breast or mammary model. With this one, we have a listing of the structures that you need to know that's separate from that in the lab manual because the numbers have changed on this model. We need to know the lymph nodes like you see out here. This is just off the top of my head. The actual lactiferous glands that we see here that look kind of alveolar in structure. Then we have the mammary ducts that you see going through here, numbered 13 right there. These leading to the lactiferous sinuses, the enlarged areas that we see through here. Those leading to the lactiferous ducts that then come together in the nipple, surrounded by the areola. Okay, and then this slide just reminds us that certainly in this last section we will be using the torso models, both for the reproductive structures, but also for any of the other structures. For instance, here are the adrenal glands. You can see the pancreas there for endocrine system as well. This is also a reminder that not only do we have the plaques for the female and male reproductive system, but we have the 3D representations of them from the torso model, so you need to be able to recognize structures both on the surface, if you will, as well as the mid-sagittal cut. So for instance, this is the vas deferens coming around here like this. This is the bladder up here. When we see it in side view again, we can see the bladder here. You can see the seminal vesicle along the side here, things like that. One other thing to note, when we see the mid-sagittal cut of this torso model, male reproductive anatomy, it does a better job of showing the three different sections of the urethra. So what we have is called the prostatic urethra through here. The membranous urethra is just as it's exiting the body cavity right here. And then we have the spongy or the penile urethra right here. The flat plaque goes through it fairly well, but it shows the prostatic. It shows the spongy and penile, but the membranous isn't really shown well there because they have the prostate extending down much more inferiorly than this model. Alright, so let's move through to some of the histology. So this is the ovary slide of the cat. We also have, I believe, an ovary slide of the rabbit, which you can look at in comparison. So again, this is going to be cortex out here. Medulla is going to be in here. Cortex is where we're going to find the follicles. We see the primordial follicles through here. All of these small ones. Here's primary follicles here. And these are good secondary ones here with the fluid-filled antrum. There's the oocyte. There's the oocyte there. You'll also see what's called atriatic follicles, those that did not complete the developmental sequence and actually have a little spot of blood inside of them. We see here and over here. So again, here are primordial follicles through here. This one right here is on the transition of becoming a primary follicle. You can see cuboidal shaped follicular cell surrounding it. It's getting a little bit larger. Here's a good primary there's a good primary over to the right side of this one, but mainly a secondary that we see here. And again, we see the fluid-filled antrum. We see the oocyte here. We see that again, the pellucidum right there. We see, you'd have to get up much closer to be able to see like the corona radiata, etc. And then we see what are called the granulosal cells or the theca cells. And those are really basically the same cells, just kind of different developmental stages. This is another one showing medulla down at the bottom. Bunch of follicles out here. Again, here are two secondary because they both have the fluid-filled antrum. This is much smaller. So of the two, we would call this one a mature follicle and this one just a secondary. We can see some atriatic follicles again over there. This just shows a good zoom up of, again, a secondary follicle with our oocyte here. Here's that pinkish pellucidum we were talking about here, the corona radiata, which is going to be this first layer of cells through here, and then the cumulus ophorus, which is the cloud of cells, which are basically, like we said, granulosal or thecal cells. This is a corpus hemorrhagicum that we see here with the blood on the inside. I haven't seen too many of these in the slide, so I wouldn't worry about identifying them in the slide. This whole structure here is a corpus luteum. So a solid ball of cells, like we said, that secretes progesterone in order to maintain the uterine lining. And some atriotic follicles again. When we get to the uterus slide, there are two uterus slides because we have to identify two different stages. In both of those, you need to identify endometrium and myometrium. Endometrium is going to have the endometrial glands, or the uteral glands that you see here. And then the myometrium is going to be the solid pink smooth muscle where those are absent. Maybe a little bit over to the right-hand side right here. So we'll see that in both of the uterine slides. But we also need to identify the phases. And in order to identify the phase, kind of like the pre-ovulatory or post-ovulatory phases of the uterus, which is also called the proliferative and the secretory phase, or the secretory phase, depending on where you're from, with the uterine glands in the proliferative phase, they tend to look smooth, much like this one is. And when you look at them in the secretory phase, they have this kind of wavy border to them. Very well seen right here. Okay, so this is the secretory phase here. And here's myometrium. Again, the only spaces you would see in here would be like blood vessels cutting through instead. So you need to identify two layers, endometrium and myometrium. You need to identify the uterine glands, and then you need to figure out whether it's in the proliferative phase or the secretory phase. For the male, we've got two slides. We've got the epididymis and then we have the testes. With the epididymis is the comma-shaped storage organ that is on top of the testes. And when we look at them, what we have is, commonly this is a pink or purple stained slide, and it has a fairly clear lumen to it, a fairly open lumen. You may see microvilli, so it may have a somewhat fuzzy border if you really zoom up on it on high power. But the main part of the lumen, the open space in the center, is pretty open and clear. When we look at the seminiferous tubules within the testis, again, here is the lumen. These are cross sections, right? And in here we see the tails or the flagella of the mature sperm cells. Now, the lab manual and the book, of course, talk about the different developmental stages of the sperm. And if we zoom up a little bit, here we're seeing just kind of one section, and here's one section of a seminiferous tubule, and so you'd have immature cells out here, like spermatogonia they're called. And then as we go closer and closer to the lumen, you would have more and more mature sperm cells. So you go primary and secondary spermatocytes, and then spermatids, and then spermatozoa once you're in the inside here. Now realistically, if you can identify the spermatozoa towards the outside, like up here as well, and then just the spermatozoa on the inside with their flagella sticking in, that would be fine. And then these as seminiferous tubules. The other thing that we see here is outside of the seminiferous tubule, we see areas that we have cells, and these cells are what's called the interstitial cells, and these are the ones that are responsible for secreting testosterone. So those are also important in this slide.